The major discoveries of the study include antimicrobial and antibiofilm activities of vitamin D and vitamin K1. Vitamins D and K1 exhibit antimicrobial activity against tested gram-negative bacteria of Acinetobacter baumannii, Klebsiella pneumoniae, and Pseudomonas aeruginosa at dosages ranging from 625 to 1250 and 2500 to 5000 microgram per mil respectively. Both vitamins were also found to significantly reduce bacterial surface adhesion and eradicate mature biofilms of the selected bacterial isolates, and were confirmed by a notable suppression of biofilm encoding genes CUSD, BSSS, and PLA by 66 to 100% in A. baumannii, K. pneumonia, and P. aeruginosa respectively. Next, the hypothesis that leads to this study is the prominent increase of antibiotic resistance in a wide range of pathogens which is significantly associated with biofilm formation, particularly gram-negative bacteria. Furthermore, usage of pharmaceutical supplements may enhance the treatment of antibacterial agents due to their antibiofilm properties against the bacterial strains. There are three main objectives of this study, which include to evaluate the antibiofilm activity of both vitamin D and K1 against common gram-negative organisms, which are known for their biofilm-forming abilities and antimicrobial resistance. Second, to assess the impact of vitamins D and K1 on the initial bacterial attachment and maturation of biofilms developed by the selected gram-negative isolates. Next, the research aimed to investigate the effects of vitamin D and K1 on the expression levels of biofilm encoding genes in the tested bacterial isolates.
There are three major methods. The first method is antimicrobial susceptibility testing. The purpose of this method is to determine the resistance of bacterial isolates to various antibiotics. The Kirby-Burr-Diss diffusion method was used according to the Clinical Laboratory Standard Institute (CLSI) guidelines to assess the susceptibility of A. baumannii, K. pneumoniae, and P. aeruginosa isolate to different antimicrobial agents. Next is biofilm formation assessment. This method aims to evaluate the ability of bacterial isolates to form biofilms, which are communities of microbial cells that adhere to surfaces and are often associated with increased antibiotic resistance. Lastly, minimum inhibitory concentrations, MIC6 determination. The purpose of this method is to establish the lowest concentration of an antimicrobial agent that inhibits the visible growth of bacteria. The MICs of vitamins D and K1 were determined using broth microdilution plates according to the CLSI protocol. There are 5 major results. First, high prevalence of biofilm producers. Out of 130 gram-negative isolates, 93.2% were confirmed to be biofilm producers with varying degrees of biofilm formation capacity. This result indicates that biofilm formation is a common trait among these pathogens, which is problematic as biofilms are more resistant to antibiotics and can lead to the persistent infections. Next, antibiofilm activity of vitamins D and K1. Both vitamins significantly decreased bacterial attachment and eradicated mature biofilms in the tested isolates. The MICs of vitamin D and vitamin K1 were established with vitamin D showing MICs of 625 anti-1250 microgram per mil and vitamin K1 showing MICs of 2500 to 5000 microgram per mil against the selected isolates. Third, antimicrobial resistance. The isolates exhibit high resistance rates to several antibiotics including B-lactams, ciprofloxacin, amikacin, and gentamicin. This finding underscored the severity of the antimicrobial resistance crisis and the urgent need for alternative therapies. There are 5 following gene expression analysis. The study found that vitamins D and K1 significantly reduced the expression of biofilm encoding genes CUSD, BSSS, and PELA in A. baumannii, K. pneumoniae, and P. aeruginosa respectively. This reduction in gene expression provides a molecular explanation for the observed antibiophilic effects of the vitamins. Lastly, potential adjuvant therapies. The results highlight the potential of vitamins D and K1 to be used as adjuvant therapies to increase the efficacy of treatment for infection caused by multi-drug resistant strains and biofilm-forming phenotypes.
Next, from my understanding, there are 3 ways to interpret the results differently. Firstly, the phenotypic and molecular validation of gram-negative bacterial isolates. Out of 130 gram-negative bacterial collections, majority were identified and confirmed to be A. baumannii, Klebsiella pneumoniae, and Pseudomonas aeruginosa based on phenotypic and biochemical characteristic. Further validation was conducted using multiplex PCR targeting these two genes, confirming the presence of these genes in all phenotypically identified isolates, thus validating the isolate's capabilities in biofilm formation. Secondly, transcriptional response of biofilm regulatory genes to vitamins D and K1. Both vitamins inhibit biofilm formation by affecting bacterial adhesion and possibly altering the EPS matrix production. Gene expression studies showed significant down-regulation of the genes under both vitamin treatments compared to the untreated cells. This indicates suppression of biofilm-associated genes. Thirdly, therapeutic potential of vitamin D and vitamin K in biofilm-related infections. Both vitamins are beneficial to intestinal microbiota. As for vitamin D, it affects bacterial colonization by stimulating antimicrobial peptide production and maintaining gut microbiota balance. It is also capable of inducing antimicrobial peptide secretion and reducing inflammatory cytokines. Meanwhile, as for vitamin K, it helps in managing inflammatory bowel disorder. In conclusion, this research demonstrates that vitamins D and K1 exhibit antimicrobial effects against the tested strains at therapeutic doses, inhibiting biofilm attachment and disrupting mature biofilm structures. This inhibitory action was spotted by a significant reduction in the expression of genus-specific biofilm-associated genes. The potential of these vitamins to combat biofilms suggests promising applications in clinical treatments against these pathogens. Co-administration of antibacterial agents with these vitamins may mitigate or reverse resistant mechanisms of bacterial strains, although further investigations are necessary to evaluate their efficacy in vivo.
Sources such as journal articles, reports, and databases are used as supporting evidences, including the correlation between low serum vitamin D and chronic dental caries in children, the role of vitamin D in fighting bacterial infections caused by Staphylococcus aureus, Pseudomonas aeruginosa, and Helicobacter pylori, the potential of vitamin D to disrupt dental plaque, and the use of vitamin K3 as effective biofilm inhibitors against staphylococcal-associated surgical site infections. The limitations of the research, including limited scope of bacteria, where the study focused on gram-negative bacteria, which are A. baumannii, K. pneumonia, and P. aeruginosa. Thus, it is possible that the findings do not apply to different bacterial species or strains. Another limitation is lack of in vivo validation, where the study conducted are based on in vitro experiments, which the result may be varied on in vivo experiments. Lastly, the potential for resistance development, which is the study did not address the common problem with antimicrobial medicines, where bacteria will eventually become resistant to the vitamins.